This video is on the development of the B cell in the bone marrow. The B cell starts its path in the bone marrow and then travels to the spleen to complete its path. The B cell grows from the HSC. The HSC divides into the common lymphoid progenitor to the Preprob B cell, where it is committed to become a B cell. The further development of the B cell will be influenced by the niches in the stromal cells in the bone marrow. As the B cell travels through its pathway, the cell surface markers change. These changes will define where the B cell is in its pathway. The first step is becoming a Preprob B cell. To do this, it starts to express CD45R surface markers along with RAG1 and 2, TDT, IL7R, and CD43. What the Preprob B cell doesn't have is antibodies. It moves from the stromal cells that secrete CXCL12 to the stromal cells that secrete IL7, since it now has the IL7 receptor. The next stage is the early prob B cell. In this stage, DJ heavy chain recombination starts. The cell also starts expressing CD43 and CD45 and class II MHC. The big move here is the joining of the heavy D domain and the heavy J domain of the immunoglobulin. Once this happens, CD19 and CD40 are expressed, and the heavy V domain is joined to the developing immunoglobulin. The cell has now entered the late prob B cell stage. The essential point is the expression of the Ig alpha, beta, and the CD19 to continue the pathway. The next step in the B cell development is the pre-B cell stage. Here it starts as the large pre-B cell. It maintains the 4 CDs, CD43, CD45, CD19, and CD40, and the class II MHC. But the RAG1-2 is no longer made, although the TDT is present. The surrogate light chain is produced, and the immunoglobulin formed is known as the pre-B cell receptor, or pre-BCR. This receptor becomes embedded in the membrane. If this does not happen, the large pre-B cell will die. This is the first checkpoint for the proper development of the B cell. When all the properties of the large pre-B cell have been produced and embedded, RAG1-2 comes back, and IL7R and TDT decrease as the cell enters the small pre-B cell stage. CD43 is no longer expressed. Now the rearrangement of the kappa and lambda light chains occurs. Once completed, the pre-BCR decreases and the IgM receptor stops any further production of the pre-BCR. Once the IgM is embedded in the membrane, the small pre-B cell passes the second checkpoint and can now complete its transformation in the bone marrow. This final step is the immature B cell. IgM is found in the membrane, RAG1-2 decreases, and TDT, CD43, and IL7R no longer are expressed. These cells undergo negative selection by being exposed to self-peptides. Only the cells that do not have a high affinity for self-peptides are allowed to live. If the B cell does have a high affinity, it has the opportunity to edit their receptor. If it does not, it will die. The cell is ready for transport to the spleen and maturation. This complex pathway is for the development of the immature B cell. Its journey, however, is not over.